Hey guys, welcome back to this MCAT biology series where I'm going to be covering bacteria and viruses today, and I also have a MCAT style practice question. Okay, let's begin. So, if you watched my last video, we covered the eukaryotic cell. Today, I'm going to be covering prokaryotic cells against eukaryotic cells. So, on the MCAT, really the main thing about prokaryotic cells is that, for one, they're not eukaryotic cells, so they're not us. And for two, they really relate to bacteria. So you're going to get a lot of questions about bacteria, not about bacteria specifically, but you're going to use them in lab experiments and infection studies and things like that. Let's go over the basics. So I'm going to just fold this real quick. Oh, little camera wobble. Eukaryotes have a number of really interesting properties. For one, they have chromosomes and they have multiple stretches of DNA. So this is DNA. They have multiple stretches of that. Another thing is that they can be unicellular, so just on their own, or they can be multicellular in a community, just like our muscle cells or bone cells or really anything like that. Eukaryotic cells also replicate via mitosis, and they can also undergo meiosis. So mitosis is just copying and then splitting, and you have two identical daughter cells. On the other hand, meiosis is sexual reproduction. So that is when you create from one cell to two and two to four, and you create either a sperm or an egg. And if that's confusing to you, that's okay. I'm going to cover that in a future video. This section right here, I'm going to hold off on, and I'm going to use that to compare to prokaryotes. Okay. If you look at the other side, we see a prokaryote, and this is a prokaryotic cell. Really, when I talk about prokaryotes, though, I'm mostly going to be talking about bacteria. There is another type of prokaryote called an archaea, A-R-C-H-E-A-E or something, but you're really not going to get content about archaea on the MCAT. Just know that they're archaea, they're not bacteria, they're not viruses, but they're also not animal cells, and they have some interesting properties. Okay, so for starters, prokaryotes, and mainly bacteria, only have one circular DNA. So instead of having multiple chromosomes like that, they actually just have one circle. So it's a single DNA. As well, they have these little tiny things called plasmids. What is a plasmid? It kind of just looks like the same thing, right? And that's what it is. A plasmid is just a small version of this single DNA. I like to think of it as like a USB drive. Bacteria can share this with each other to gain or lose function. So, important for us, future doctors, and also people who are studying for the MCAT is why plasmids can impact health. Sorry, the camera's just shaking a bit there. So there's a bunch of different types of important plasmid. One of these, for example, let's call this a virulence factor. What does that mean? A virulence factor is something that the bacteria can get and become infectious with. Okay? That's a virulence factor. It causes the bacteria to become infectious or more dangerous or something like that. On the other hand, right here, we can have drug resistance. So, if you've ever heard of MRSA, that is methicillin-resistant Staphylococcus aureus. Okay, that is the methicillin-resistant version of a staph infection. Methicillin is an antibiotic. So they found that certain antibiotic resistance are found on these plasmids, which is why bacteria like to pick them up and trade them around. Okay, so we have a prokaryote bacteria with one circular DNA and plasmids. They don't need to have plasmids, but they can. Next thing, we're going to compare the shape. So a prokaryotic bacteria has a cell membrane and a cell wall. Okay. The cell wall allows the bacteria to do a bunch of different things. It can have drug transporters, it can have certain receptors that do things. You're going to see different questions maybe about what the use is. All you really need to know is that the cell wall makes something either gram positive or gram negative, okay? Gram positive and gram negative refers to a test we use to identify what bacteria is what. So just know that cells can either be gram positive or gram negative, and that's based off of the cell wall. That cell wall can either be composed of a thick layer or thin layer of something known as peptidoglycan. But that is a little out of the scope of the MCAT, and I really don't think they would ask you about that. Another important thing is that they have no, let me do this in a different color, membrane-bound organelles. So everything in here is just like a big soup. It's like one stew. So here we have compartmentalization of the nucleus, the mitochondria, the endoplasmic reticulum. In here, everything is together. There's no compartments. As well, they are unicellular always. So one bacteria is always going to be on its own. During an infection, there's going to be a lot of bacteria, but they're not connecting together like our cells are. Okay, next step. Bacteria can replicate really, really fast through a process called binary fission. Okay, fission, if you want a way to remember that, means it's the splitting of something. Like in a nuclear reaction, you can have nuclear fission where one atom splits into two. Here we have one bacteria copying itself and then turning into two, really quick. That's in contrast to mitosis and meiosis, where mitosis is much slower and meiosis generates 4 cells that are not identical. And then finally, we have actually a really common, a commonality. We have a similarity. Both prokaryotes and eukaryotes can become infected with viruses. So when a bacteria becomes infected with a virus, it is known as a bacteriophage. This one's easy to remember. Bacteria, phage, maybe you don't know what that is, but bacterio, something that is infecting the bacteria. Okay, this bacteriophage can be lysogenic or dormant. What does that mean? Lysogenic? Well, anything that's genic means creating, and lys, that term is for breaking apart. So like Lysol breaks. So something that's lysogenic means it's breaking the cell. So this bacteriophage will come into the bacteria, recreate, break the cell, and leave. Lysogenic. Dormant, meaning it injects its DNA, and the DNA just joins up with that circular DNA of the bacteria, and it just sits there. On the other hand, in eukaryotes, and I'll just talk about humans, for example, we get infected with viruses. Here I drew SARS coronavirus, which we all know about. So these are the main differences between prokaryotes and eukaryotes. This is what I would know for the MCAT. I wouldn't know this in super detail, but just know these factors. However, that talk about viruses is leading me into a good segue for the next section, which is viruses. So how do viruses infect cells? Now, this process is important for the MCAT because they're not going to ask you specific content questions. They're not going to say, what's the second step of a viral infection? They're going to give you some sort of question, and they're going to use a virus as part of that question, and you're going to need to understand the steps to get the right answer. So, bare bones, down and dirty, viruses. There's positive sense and there's negative sense. There are also single-stranded and double-stranded, but that's not something that you're going to need to know really for the MCAT, or if you do, they'll explain it. Let's start with a positive sense. Positive sense viruses have either DNA or RNA that they inject into the cell. Okay? Viruses can do that because they connect with the cell, they're intaken, and they're actually pressurized. So when a little hole opens, it pops and shoots everything in. So the positive sense virus is shooting in its DNA. The cell doesn't know what to do with it. It's just a cell. It doesn't have a brain. So it thinks, oh, I have DNA or RNA in my cytosol. That must be mine, right? And because of that, well, I got to do something with it. So let's recreate it and let's make some proteins, because DNA and RNA make proteins. And if you don't know about that, that's fine. I'm going to cover that later. But just know for now, DNA and RNA are used to make proteins. So the cell gets this viral DNA or RNA and it says, okay, well, it's mine. Let's do something with it. So it recreates it. And so now it recreates a ton of viral DNA, a ton, and it makes a ton of that, wrong color, makes a ton of that viral protein. And that viral protein are the balls, those little ball things that the coronaviruses are in. That's called the capsule. So we see strands of DNA. We see the capsule. Well, what left is there? But let's get out of here. Break the cell open. Boom. More viruses. We can redo that all over again. Boom, more and more and more. So you understand why viral infections can quickly run out of control, because they hijack the cell. They use the cell's machinery to make more of themselves, and then they break the cell open and leave. That's a positive sense. Positive's good. It's easy. A few steps. That's it. Negative sense. Negative. It's like backwards. It's like a 1 and a -1 are like opposites of each other. So in a negative sense virus, it's still DNA or RNA, and it still shoots its DNA or RNA in, but the DNA or the RNA is actually reversed. Now, that might not sound like a big deal, but for example, if we think about the English language, this is what a Z looks like, right? Okay. If I gave this letter right here to a kid and I asked them, what does that say? They would have no idea. And to an adult either, they wouldn't know what that letter is. So the orientation is important, and we'll cover that later. But negative sense is backward. So what do? They use a special protein that they have to turn themselves forward. And I forget what the name of that one is. It's I have it in my notes somewhere. I'm not sure. I don't think the name is really important, but you'll see that it reverses it. And then when it's reversed, when the DNA is in the right direction, it's basically just positive sense. Meaning, make more DNA, make more protein for those capsules, break the cell open, boom, infection. In both cases, the virus is entering the cell, which is called the host cell, using its inbuilt machinery, because all viruses have is the capsule and the DNA. They don't have anything else. They are freeloaders. They are mooches. They are stealing your fries and leaving. So that is how viruses work. Okay, we know that viruses can cause infections, but did you know that bacteria can also enter our body and cause infections? So I made an MCAT style question. I'm going to read it to you. Researchers discovered that P. bananas, a gram-positive member of the Prokaryotes family, binds to the P34A receptor on the surface of chicken muscle cell. Binding has been shown to increase the amount of P. bananas cell wall inside the chicken cell. What does that mean? I'm confused. If researchers created a knockout version of chicken muscle cell delta P34A, which graph of internalized cell wall amount is most correct? I want to quit. I want to give up. I actually have no clue what's going on and my test. Okay, that was dramatic. Let's try and figure this out. MCAT's really hard. They're going to give you a lot of questions that you don't know about. And if you don't come from a science background, you're going to freak out. Even if you come from a science background like me, you're going to freak out. I know I did. I would read some of these, especially under time, and I'd be like, bruh, like, what? But let's relax. Let's remember we're smart, or kind of smart. Let's break it down. Researchers discovered that blah, 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 gram-positive, blah, 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 blah. Gram-positive. Let's do this in a different color. Okay. We know gram-positive, gram-negative, that refers to bacteria. So we're dealing with some sort of bacteria, right? Member of the prokaryote family binds to the P34A receptor. Okay, receptor. That's some protein or something. Okay, on the surface of a chicken muscle cell. Binding has been shown to increase the amount of P banana cell wall inside. So when the bacteria and the receptor connect, there's more cell wall on the inside. What does that mean? How could that be possible? Well, this is one way to think about it. You have your bacteria. You have your chicken. And let's say that this is the receptor. And we know that, let's see, binding increases the amount inside. Binding means that the proteins are touching. So I don't know how that works, but we know that after that happens, there's cell wall inside the chicken. We don't know if it's full, we don't know if it's broken, but there's cell wall. How does that happen? Well, the only way that can happen is if the chicken cell basically takes that in. So it seems like somehow that binding brings the bacteria in. Maybe it's used to defend the chicken cell against the bacteria, but we know binding has been shown to increase the amount of cell wall inside the chicken cell. Binding brings it in. Now we understand what they call the mechanism. This is one possible mechanism, but let's just work with this for now. If researchers created a knockout version of chicken muscle cell, delta P34A, what graph of internalized cell wall amount is most correct? Okay, I'm freaking out again. What is a knockout version? Knockout, or KO, means removal of blank. So they removed P34A. Removal is often shown as that triangle called delta. If researchers created a knockout version of chicken muscle cell, P34A, meaning the cell's missing this, which graph of internalized cell wall is most correct? So let's just draw this out again. Now we have the chicken cell wall, but there's none of this receptor. Well, we know that when the receptor is present, the bacteria comes in. But removal, well, that probably means the bacteria can't get in, huh? So I don't know if that's true. Maybe the bacteria uses a different receptor that we don't know about. But for now, with the content that we have, the answer of the least assumptions is it just comes in. It just comes in with the receptor. It doesn't come in without it. So let's look. What are these graphs? Graphs of internalized cell wall amount. So the positive represents the receptor being there. The delta represents no receptor. So let's think. When there's when the receptor is present, we should have it in. Okay, so this alone doesn't tell us anything. And when it's gone, we should have no cell wall in. So let's look when this is big and this is small. So this isn't right. This isn't right. Positive, the cell wall's inside. Negative, there's not a lot of cell wall. This is just reversed. So, boom. 520 plus. Right there. Boom. Harvard Medical School. But really, it's that's the type of thinking that you want. One important thing to note that threw me off is if it was delta, you would expect no entrance, right? But in science, there's always going to be a little bit. So don't freak out if you see this and you're looking for one that has nothing, because you're not going to find that. It's okay to have a little. All right. Well, with that being said, I made a little list of the vocabulary that you should know. I'm not going to explain it all because you can look it up and it's boring to me. But for bacteria, these are the words and things I would understand. And for viruses, these are the words and concepts I would understand. All right. Well, I appreciate your time and I hope that this has helped you. Have a good day.